Our last talk will be from Hanyu Zheng from the Bailey Lab. Hanyu will be talking to us about impacts as synthesis conditions on membrane protein incorporation into nanodiscs.
Thank you, Brendan, for the introduction. Again, my name is Hanyu. I'm from Bailey Lab here in University of Michigan. Today, I'm going to have a talk about my research on membrane protein incorporation into nanodisc and how the synthesis condition can affect that. So this is my outline. We'll discuss briefly about the membrane protein and why we use nanodisc to study them, and go into my result. So membrane protein precipitate in many important cellular processes such as signal transduction and transfer molecule in and out of membrane, which is why that even though only about 30% of human proteome are membrane proteins, about 60% of the pharmaceutical targets out there are actual membrane proteins. And to understand them better, one might study their function and their actual structures. But for structure-wise, if you look into protein data banks, only about 3% of the protein out there are actually membrane protein structures. And you can see that there's a gap between how much we want to target them and how much we really actually know about their structures. And this is likely due to the hydrophobic effect. The hydrophobic region on the membrane protein, which could when we remove them from their native environment, could cause them to misfold, unfold, or aggregate. At the very least, we can cause them to lose some sort of function. This is why that many researchers has developed some sort of membrane mimetic, such as my cell here, or just suspend the membrane protein in some sort of detergent kind of solutions. While this detergent gives us a great solubility, the dynamic between the detergent and the original lipid bilayer is somehow different, and it's harder for us to study the actual original membrane protein functions. And there's cells, which utilize a lipid bilayer capped by some sort of detergent, shown here. It gives us a more native environment. However, it's harder to put some sort of purification tag for any downstream analysis for our membrane protein. And there are liposomes, which is very easy to assemble. However, they are very polydispersed. It's hard to control the lipid composition of the liposome. It's hard to control the size of the liposome. The membrane environment for the membrane mimetic actually are causing the membrane protein to take on different functions. Therefore, the polydispersity of the liposome kind of hinder our study for their functions. And there's nanodisc, which I'll be focusing today. It has monodispersed sizes, as we can easily control the lipid composition, and we can easily tag them. But it's fairly new technology, and many things needs to be studied about them. So this is a closer look at the nanodisc diagram. We have a lipid bilayer, similar to bicell I was talking about earlier, and we have some sort of scaffolding to make the nanodisc compose together. And the scaffolding we'll be using today is some sort of protein called membrane scaffold protein. They are tunable sizes, and we can tag them with biotune tag for further strep-avidin purification. And this nanodisc has been shown to stabilize protein aggregation inside an aqueous solution for about 30 days at 4 degrees C. And this is our current workflow in our Bailey lab to synthesize nanodiscs and analyze what kind of proteins are in the nanodisc. So we're starting with 4 materials. We have our MSP, the scaffolding, the lipid that we want to put in. We have our protein sources. We have our detergent conditions. Once we suspend everything in the detergent condition, we'll remove them, which form the nanodisc. And subsequently, we'll use strep-avidin column to purify the nanodisc by selectively binding to the biotune tag that's currently on the MSP. Then we'll disassemble the nanodiscs, purify them, by removing all the salt, the detergent, and the lipid that could be harmful to mass spectrometry. And we do a bottom-up proteomic workflow to analyze what kind of protein that's initial here. And like I mentioned when I mentioned liposome, altering the memory mimetic condition, altering the lipid environment in the memory mimetic condition has shown to be changing the memory protein function. And the goal for us is to study that how the synthesis condition that we mentioned before can affect memory protein incorporation into our nanodisk samples. With that, I'll go into first part of my results. So this is, since we're talking about synthesis condition, I will be focusing only this part of the workflow. Previously, by Roy et al. Has shown that when we're changing the different lipid composition of the nanodisk classes, and the memory protein classes they extracted have been shown significant different. However, we believe that not only the lipid composition itself can change the lipid environment, but also things such as detergent that's suspended, everything, and also the MSP sizes / nanodisk sizes could also change the lipid environment. And the goal for us is to have a set synthesis condition that allow us to further selectively enrich the memory protein or memory protein classes that we desired. And this is the zoom-in version of the starting material. In this experiment, we'll be holding two constant, which is the protein source, which will be the memory protein extra, and we're also holding the detergent constant, which will be cholate. What we're changing is the nanodisk sizes by changing to different MSP, which I'll be talk about in depth slightly later, and we'll be having 4 different lipid composition. And since not all of us are lipid experts, I'd like to dive a little bit into the lipid background. These 5 lipids, the 5 groups we'll be using today. We have phosphatidylcholine, phosphatidylethylamine, phosphatidylserine, phosphatidylinositol, and a sphingosine lipid called sphingomycin. And as you can see, as a natural composition here, the phosphatidylcholine are actually dominating the natural composition of the lipid. Therefore, most of nanodiscs will be PC-based. So this are the lipid composition we'll be using today, 4 of them. The first one will be 100% PC, the second one and third one will be a mixture of PCPE and PCPS, respectively. And the last one we call ER is inspired by the lipid composition of natural endoplasmic reticulum in human memory. And then we have 2 different sizes of MSPs. The larger MSP will be called MSP1E3D1, and the smaller will be MSP1D1. And these symbols will continue throughout the slides, help us distinguish between the bigger and smaller MSPs. We also have 2 controls. We have a positive control, which is our starting material, and the negative control, which is nanodiscs without the biotin in them. And when we're running through strep-avidin column, nothing should theoretically bind, and nothing should not theoretically bind. Therefore, it should be fairly clean. And just to mention our sample name structure, the sample name with first part will be our MSP and second part will be lipid. For example, this sample name was telling us that it's made of larger MSP and it's made of some sort of mixture between PC and PS lipid. So the intensity that we use in the study will be max L of Q. Just a quick go through about how it's calculated. We'll first have our peptide intensity and their spectrum match between our spectrum and the database, and we'll construct a max L of Q intensity by comparing the peptide intensity between different samples. A different way to visualize this is, for example, we have sample A and sample B. You can see the red guy is taller in the sample B, but not really taller than the normal guy in the sample A. And this normalization allowed us to compare the protein density between different samples. And we also have some problem with our negative control, which have very high intensity but very low protein ID counts. And along with, we found that the protein with the most highest intensity in our samples are actually consistent between our samples and negative controls. This is telling us that a few protein that's sticking around one reason for another, by non-specific interactions are detecting our sample intensities. So we decided to remove them and pull everything together as our normalized intensity. This is our the total mean graph over 3 samples. Not much of surprise there. We'll have our positive control slightly higher than our bigger nanodisk, which is also slightly higher than a smaller nanodisk. The bigger side does allow it to incorporate a bigger protein or incorporate more proteins. And we can also break each of these bars into protein classes, something like this. This is just the top 6 memory protein classes in intensity out there. And I think a better way to do this is we can compare each bar in the samples with the positive control to show the enrichment of the memory protein the nanodisk actually give us. And this is our bigger nanodisk. And the first thing that we see here is that our cytosolic protein in our nanodisk content has decreased. And we also see that some memory protein classes, such as receptors and transporters, are increased by quite a lot. And the same thing also happened in our smaller disk samples. And this intensity actually is slightly, although not significantly higher compared to our bigger disk samples. So in conclusion, we found that some memory protein classes are enriched in membrane, in our nanodisk membrane, such as transporter receptors, and we see a decrease in cytosolic protein. And we believe that this increase in intensity for transporter and receptors are not unique. So we take a slightly deeper look at all the transmembrane protein that's in our sample to see if they are enriched. So this is bigger nanodisk, just one of the lipid composition. We can see all the golden dots here are the transmembrane proteins, and we see the top 50 most intense protein consists mostly a transmembrane protein. This is not, and this is comparing to about 30% overall protein IDs over average samples. And this is also happening in our smaller nanodisk, although slightly lower in the percentage-wise, still about 59% of protein are transmembrane proteins. And we actually can plot this for our not only top 50, but up to all the top 750 samples. So the red line dot here is the average memory protein count in the samples, which is about 30.6%. And we can see it's about 20 to 25 increase over the line. So at top 500, the memory proteins, transmembrane protein actually have higher number versus the non-transmembrane proteins and cytosolic protein in the sample. And the conclusion for this part is that we see nanodisk are actually very good at enriching transmembrane protein overall. As for the conclusion, in this presentation, hopefully I show you the nanodisk's ability to actually enriching certain membrane classes, and specifically transmembrane protein or some integral proteins. And for our future direction, we can do, we actually propose to examine different type of detergent. The thing that we're not changing in this experiment today, we'll try to minimize the detergent's effect on the suspension of membrane protein. Hopefully we'll get a more native-like protein in our nanodisk. And our end goal is to leverage the nanodisk synthesis condition to enrich memory protein classes of our choice. With that, I would like to go to acknowledgement slides. Thank you, Dr. Bailey and Bailey Lab, for supporting me. I'll take any questions.
Thanks for the talk. I know that's a lot of work. This is not directly about your talk, but I'm curious about something with these nanodiscs. Some proteins I'm thinking about things like ion channels that are multi-components, and then they can perform functions, like opening up a pore to allow ions through. Has anyone been able to recapitulate that in a nanodisk and show that they can get sort of a functional multimeric protein in these guys?
I'm not sure about multimeric protein, but I've seen study where some integral memory protein have retained their function. Whether it could be enzyme or it could be ion channel, like you said.
Yeah, I think the technical applications of that kind of stuff are huge if you can do that kind of thing.
I had one quick question. I know you and I have talked about this data a lot, so I want to ask like a general sort of future thinking question. I mean, when you started generating this data, I think what was surprising to me is just how many cytosolic proteins seem to be coming along for the ride here. And there's always this open-ended question about how much of that is just sticky protein, how much of that is like real information content about what is maybe from the cytosol associating with the periphery of the membrane. I wonder if you've thought at all about how to disentangle that, maybe with future data or maybe within your current data set?
Well, I believe that if it's true that cytosolic protein are sticking to like associated such as like membrane protein, those protein could have some information for us, and those I believe they are good to keep. As of how to fix it, we could try a little low dose of maybe anitosol could interrupt the binding for the cytosolic protein. If it's fully incorporated into nanodiscs, those proteins should still continue to be in nanodiscs after we disrupt those loose interactions. And for those proteins with strong interactions, again, it's also a good data point to have.
Okay, let's thank Hanyu again.